that
Recording in progress.
initially we discussed how the cell signaling will be done when there is a signal and how your cell membrane will respond and how that signal can be amplified. And then we discussed, beside the example involving in the second messenger, how without involvement of second messenger, okay, this question should be without using second messenger. How can you, how receptor can alter the activity of the cell? Can you tell me now? Without using second messenger.
Without using the second messenger,
Hmm.
it can act as a membrane. No, not as a membrane, as an enzyme.
Hmm.
For it can act as an
Membrane
enzyme
bound
and
enzyme, yes. Membrane bound enzyme.
membrane
Directly it
bound
can,
enzyme.
yeah.
Yeah, and it can create a
Okay, your
cellular
answer, this
charge.
is half answer. This is half answer. You must say that it can act as a membrane bound enzyme and can generate response directly. Okay,
Yeah.
it does not need any,
Okay.
it does not need any of the intermediates.
Okay.
Yeah, next.
And it creates a ion pathway,
Ion channel, yes.
ion channel, which alters the impulse of the
Potential of the membrane.
potential of the membrane.
Yes. And this is how there can be direct response to the particular signal. Okay, it can directly
Yeah.
generate a response to a particular signal. But this is specific. Okay, you cannot just for they have given you these examples. These are for some of the receptors. Not all can do this. Just like you can do this for just sodium and potassium when there are signals of ions. Okay, when there are signals of ions, then you can create ion channels. In this case, there is no need to create ion channels because this is a normal signal. Okay,
Yeah.
and similarly, when you have a membrane bound enzyme, particularly just like the insulin receptor. So in that case, there will be membrane bound enzyme. Not in all the cases, okay? Not in all the cases, you don't need any of the second messenger. You need second messenger, but there are some cases in which there is no need of the second messenger, just like this one, where you can have
Are you
ion channels. Hmm?
are you showing your screen?
Yes.
I can only see your home screen.
What? Okay. Now can you see this?
Yeah.
You are telling me very late. I don't know why I'm.
I thought you were asking me without looking. So
Okay, no problem. No sense. Okay. Yeah. Next is third one is acting as an intracellular receptor. So this is one of the ways by which your cell can respond without using a second messenger. Okay. By acting as an intracellular receptor when the initial signal passes straight through the cell surface membrane. Okay. There is no need. If you see what they have mentioned that the signal will act as a intracellular receptor when the initial there is when the signal passes there is intracellular receptor and that signal will directly pass into the membrane. Okay, there is no need of secondary messenger in this case. Okay, and in the messenger is this A or is this E messenger? What's that?
messenger, it's E, I think.
Yeah, it's E. Okay, that was correct. E.
Yeah.
Okay. For example, the estrogen receptor is in the nucleus and directly control gene expression when combined with the estrogen. So you see this estrogen, what they're saying, the estrogen receptor, it can directly control the gene expression when it is combined with the estrogen. Okay. The receptor will combine with the estrogen directly and there is no need of intermediate messengers. Okay. There is no need of secondary messenger. They can do they can directly convert that signal into the specific response. They can generate a specific response according to the signal. Clear?
Yeah.
Okay. Now this figure 4.8 summary some typical signal. This is about the previous one. I think so. Yeah. No. Where is figure 4.8? Yeah, this one. Okay, this is figure 4.8. They have mentioned some of the possible signaling pathways commonly found in the cell. What are those? Firstly, if you see, like this is an initial signal that may be light, that may be hormone, or that may be neurotransmitter. Okay. And what is this central part, this gray? What is this?
That's the protein.
Yeah, that is receptor protein. Okay. And what does it do?
What?
What is the
It
function of
changes
it?
shape
Hmm,
so that it can bind with
the signal.
it changes shape so that it can bind with the signal.
Yes. And then what will happen now? They have provided two pathways. Okay, first is this, in which this we have discussed. What is this? You can tell me now.
It transfers to a G protein
Hmm.
um
Which will
which
activate.
triggers the
Hmm.
which will activate the switch
No,
to
G protein
the
is
second
acting as a
messenger.
G protein is acting as a switch molecule. G protein activates what? The second messenger.
The
Okay.
second messenger.
Yes. And then that second messenger act as an activated enzyme, and then further it will activate some other enzyme. And the process will go on, and the signal will be amplified. Okay. The
Yeah.
newly synthesized signaling chemical is this is second messenger. Then signaling chemicals secrete if the target is extracellular. Okay. If your target is outside cell, what will happen? Once there is significant amplification of the signal, the response will be outside because you need response outside the cell. Are you getting my point? If you need the change inside the cell, so this will remain inside the cell. Okay, these amplified signals will remain inside the cell and they will make change inside the cell. But if you want a change outside the cell, then they will transfer outside the cell.
Okay.
Clear?
Yeah.
And one more thing, when this process will stop of the activated enzyme, that activated enzyme are amplifying, and amplifying again and
When
again.
the signal is received.
No. When it is written over here, it will stop
When it will
when the,
stop?
yeah, I am saying when the significant, I'm asking about this. Like
You're asking when it stops.
there are significant number of signals. Okay, there are significant responses. I am saying at what time this activated enzyme will stop its activity.
When the required
Hmm,
when the
hmm.
required enzyme is produced or
When yeah
the
yeah
reaction.
when an required enzyme is produced for that particular reaction to occur. Okay,
Yeah.
till then the enzymes will amplify. The enzyme will further amplify the signal, and it will end when a desired enzyme is produced. Okay,
Okay.
and in the extra, if the activity is extracellular, so definitely the signaling chemicals will be move outside the cell. Okay, and if there is intracellular targets, so they will move inside. Okay, like this. Are you getting this diagram till now? Are you clear with
Yeah.
this slide? Okay.
Yeah.
Then, if you see this, now they have mentioned that preformal signal inside membrane-bound vesicle, calcium ion, insulin, and adrenaline, 88 neurotransmitter. Okay. Now they are saying sometimes you don't directly release these signals. Okay, just like this. This was one of the scenarios, one of the situation in which the desired response will be released extracellular directly. But sometimes you need to convert that into a membrane bounded vesicle. Okay, because maybe that has to transfer to some other region, some faraway region, or sometimes it happens because this maybe this is hydrophilic in nature. Okay, so it cannot directly cross the membrane. Okay, so what do you need? You need a vesicle around it so that it can fuse over here and then signal can release. Are you getting my point? Sometimes your response, the desired enzyme, that may be hydrophilic in nature. that may be polar in nature. So that polar thing cannot cross the membrane. You need to cover it. Okay, your cell will,
Can you repeat that?
I'm saying for this, sometimes in this case, you see these are directly transferred outside. Okay, how they will transfer? Maybe they will be hydrophobic in nature, right? That's why they can easily
Your voice
pass
is
through
kind
the.
of glitching.
I don't know why it's happening. My Wi-Fi is, it's, I don't know.
That's okay. Can you say that again?
I'm saying, is this my screen is visible to you? Is this diagram?
Yeah, I can see it.
Okay. Uh, there's so much background noise.
I'm sorry.
Okay. Yeah. In this, I'm talking about maybe sometimes when the newly synthesized signaling chemicals, maybe they are hydrophobic in nature. Okay. So they can directly pass through the cell membrane. But sometimes the signal, the response produced may be hydrophilic in nature. So you know hydrophilic things cannot directly pass through the membrane. They cannot directly cross the membrane. Okay. So in this case, a vesicle is needed. Okay. A vesicle is needed. You will those signals will be those signaling chemicals will be bounded in a vesicle, and then they will combine with the boundary of the cell membrane, and then they can be released. They can be
Okay.
hydrophilic. There can be calcium ion, insulin ion, ADH neurotransmitter. These are some special things, okay? These are some special type of responses. So they cannot directly pass through the cell membrane. You need to bound them with a vesicle. Okay, your cell need to bound them with a vesicle. So once they are bounded through a vesicle, then they can fuse with the cell membrane, and then they can pass through the cell membrane. Okay, then they will be excreted through exocytosis. You know exocytosis? The endocytosis and exocytosis. I told you about the endocytosis.
Yeah.
What is endocytosis?
When they engulf the microorganism
Sorry?
and destroy it, and then exocytosis is when they're. I can't hear you.
I don't know what's happening. It's maybe you can rejoin. If I think there is connectivity issue on your side because on my side everything is clear.
I'll rejoin.
Okay.
Recording in progress.
Okay. So this is one of the example. I was just giving you an idea. Sometimes the response can be hydrophilic in nature, but most of the time you will see some of the special signals. Okay, some of the special signals like for the calcium ions, insulin, adrenaline. These are hormones. Okay, remember this thing for hormones, specific hormones for neurotransmitters and for ions, just like the calcium ion and potassium ions or sodium ions. You need the signal to be transported in the form of vesicles. Okay.
Okay.
Signal should be transported in the form of vesicle. Okay, for these things. And for some other things, you don't need that. They can be directly transferred. Okay, clear?
Yeah.
Okay, now then, sometimes they have intracellular targets. Okay, some of the signaling chemicals have intracellular targets, so they will produce their response in the intracellular inside the cell. Okay, and
Okay.
sometimes they need to be excreted outside the cell, so they will be excreted via exocytosis. Yes, I was asking you what is exocytosis.
It's the breakdown of
Hmm?
and it's when they release the.
It is a type of active transport. Okay,
Okay. A
and
type of active.
Yeah, it is a type of active transport. Okay. And in this, you see what is happening? This vesicle, it is releasing something outside, exocytosis. Okay. So these are, whenever we discuss about the exocytosis, we say that their cell moves the large molecules or large vesicles outside of the cell by fusing, okay, by this fusion, okay. This vesicle will fuse firstly with the cell membrane and then there will be exocytosis. Are you getting this? Let me write the definition over here. I can write it is a type of active transport in which cell move substances outside by fusion of internal vesicles with plasma membrane or cell membrane. Okay.
Okay.
Now read this and tell me what I have written and what is exocytosis.
It is a type of active transport in which cell moves substances outside by the fusion of internal vesicles with
Hmm.
the plasma
Plasma
membrane.
membrane. Now see this. What is happening over here? This vesicle is fusing, right? This vesicle
Yeah.
is fusing. And it is what is happening after
It's
fusion?
releasing
Yeah, it is releasing
the
substances outside. That is why this is exocytosis. And opposite of this is known as endocytosis. When the substances enter the cell by the fusion of the vesicle with the cell membrane. Okay, and that is also a type of active transport. Clear?
yeah.
And why we are saying there is active transport because it needs energy. You will provide energy, then this process will happen. Okay,
Mm.
that's why it is
Yeah.
active transport. Okay, now this part is clear. This whole we have done. Now this one. If we talk about this one, hydrophobic signal molecule, just like estrogen. Yeah, this is the example of where your cell can give a direct response without using. This is a direct response without using secondary messenger. Okay.
Okay.
This we have done the three ways. In the three ways, this was the third one, if you remember. This one over here. Okay. Acting as an intracellular receptor when the initial signal passes straight through the cell surface membrane. Okay, it is passing straight through the cell surface membrane. For example, estrogen receptor is in the nucleus and directly controls gene expression when combined with the estrogen. So estrogen, you see, estrogen can directly enter the cell. It does not need any G protein or the activation of the secondary messenger. Okay, it is it can directly enter the cell. Otherwise, normally what is done whenever a signal approaches to the cell, there is a receptor protein. Firstly, it will combine with the receptor protein, then it will combine with the G protein, then protein will activate the secondary messenger, and that secondary messenger will activate an enzyme. That activated enzyme will amplify the signal. Then further, there will be activation of another enzyme, and that will help to amplify the signal. This process will continue until you get a desired enzyme. Your cell get a desired enzyme, which can bring that particular metabolism
Great.
process. Okay,
Yeah. Okay.
clear? Can you repeat this?
The part where of the vesicle.
No, no. Where I am saying that Normally cell signaling, how normally cell signaling happens.
The signal, it approaches the cell and it binds with the receptor protein, and when it binds with the receptor protein, it is transferred to the G protein, and
Mm-hmm.
then the G protein acts as a switch to the secondary messenger,
Hmm.
and then the secondary messenger amplifies the signal, and it continues to be amplified by other active enzymes
Hmm.
until the desired enzyme is
Hmm. Hmm.
required. The
Produced.
desired enzyme is required, yeah,
Yes.
produced
And for a particular type of
for a
metabolic
particular,
response. Hmm.
yeah.
Okay. This is one of the direct responses. This is also one of the direct responses. If you see this, the direct cell to cell signaling is happening. This one, direct cell to cell signaling. There is one cell. This is second cell, right? This is second cell. This is first.
Yeah.
So there is direct connection. This is in what they are saying, direct cell to cell signaling by cell in contact with each other. Just like lymphocytes, they detect the foreign antigens and on another cell. Okay, this is another type. Like the, the lymphocytes, they are a part of white blood cells and they
Yeah.
help in immunity. Okay, they give immune response. So basically, they have mentioned this one as well, that sometimes there is direct cell to cell signaling in which there is no intermediate enzymes are needed. You can directly your cell will do that, and this is for some specific type of cell, just like lymphocytes. Okay, they will detect the foreign antigen on the other cell, and they can directly uh, make an action. They can directly do an action on that particular cell where there is antigen available. Clear?
Yeah.
Okay. Now there is about movement of substances into and out of the cell. This is very simple topic, very simple. You have discussed this in IGCSE as well. You can tell me what are
Yeah.
the basic processes by which substances can go inside and outside the body.
Movement to and out of the cell, there is diffusion, there's osmosis,
Hmm.
and there's active transport.
Yes. And why there is needed, why it is needed for the substances to move out of the cell or inside cell?
Um, because for which one?
For any,
For the
like,
growth
for,
of the cell.
I'm asking why it is needed for a cell to excrete things or why there is need of
They
the entry
excrete
of this,
things
some particular. Hmm,
when they are in excess or
hmm.
when they are not required
Or
or
when,
waste.
yes, or else when some substances are needed at some other position, not in the cell, just like this case,
Yeah.
this scenario. In this, signaling chemicals is needed at some other place. So in this case, they will release some of the substances. And sometimes
Yeah.
there is entry of the some of these things inside cell, just like the sodium, potassium, they enter the cell or they leave the cell to maintain the membrane potential. Okay, and there are many things which will enter the cell, just like the glucose, because when the glucose will transfer, it will be converted into ATP, and then that ATP will provide the energy. The cell need energy. So that's why for
Yeah.
survival, basically you will say for survival and for maintaining the life, the cell need to enter the substances and it need to excrete to release the substances. Okay. And there are five basic mechanisms. First one is diffusion.
Your
Hmm? Sorry, are
where
you
your
saying?
voice cut like from there. I
Is
can't hear
this?
you.
Uh, can you hear me now?
Mm, can you hear me?
Yeah, I can hear you. Your voice is clear.
I can't hear you because your voice is glitching.